First of all, my thanks to those who make this conference possible for the opportunity to show some of our work. I'd like today to take you through a journey of our efforts to get a better understanding of how birth defects, specifically birth defects of the spine and the nervous system, come to bear, and particularly under conditions where maternal metabolism plays a role. But to make sure, and I know you've just been to lunch, if you learn nothing out of this talk than this, I'd like to start with two statements. One is from Christiane Nusslein-Goldberg, developmental biologist par excellence, and she says in developmental biology, things always happen earlier than you think. And the second part is from Louis Wolpert, and it says it's not birth, marriage, or death, but gastrulation. That is truly the most important time in your life, and I'm trying to convince you of that latter fact once we get further on in the talk. Neural tooth defects are a very debilitating birth defect. They have very clear clinical significance. They can be either very mild. In fact, there are Olympic athletes that had suffered from a spina bifida yet recovered sufficiently to perform at a very high athletic level, to extremely debilitating, where people that are afflicted by a neural tooth defect are wheelchair bound for the entire life, or to simply lethal or actually embryonic lethal. It has been a great success to reduce neural tooth defect incidence by providing folate, either in the beginning of days by providing vitamins to women who wish to become pregnant, or later on in the United States since 1998 through fortification of grain products. So you can think of the breakfast cereal that you had this morning is actually a folate pill, and the other grain product sort of is more a carrier, so to speak. However, the neural tooth defect rate has not gone down to zero despite the fortification with folate. We're sitting at 1 in 2,000. And just as a last note, it is really an expensive situation. The plight for afflicted families is very high, and the cost of care can go up to $1 million. One particular cause for neural tooth defects is, in fact, diabetes during pregnancy. And it's both type 1 and type 2 diabetes, to a lesser extent gestational diabetes, but that's more of a timing issue. And what it boils down to is that there's clearly a higher risk for the embryo to come down with birth defects, for the baby to come down with birth defects. The highest incidence is actually defects of the heart and the vessel tract, followed by neural tooth defects. And what you also see quite often are defects where the bottom part of the body, from the hip on downward, is actually missing, not formed, or just a tissue mass. Maternal diabetes has another effect on embryonic development, and that is summarized by this term developmental programming. That exposure to a maternal adverse metabolic situation actually can cause the individual to be susceptible to metabolic syndrome and diabetes itself later in life. Interestingly enough, obesity, and I'm talking here about obesity without insulin resistance or type 2 diabetes, even a healthy obesity, leads to a similar spectrum of birth defects. We find heart defects and neural tooth defects that be higher in pregnancies where women are obese. There's a higher risk for stillbirth, and there's also this issue of developmental programming that the susceptibility to metabolic syndrome is much, much higher in offspring of obese mothers. And of course, you can see how that, both in the diabetic and the obesity case, that leads to a feed-forward cycle that is quite difficult to deal with. So the metabolic imbalance of the mother affects embryonic development, and it specifically affects morphogenesis. And we have some kind of phenotypic convergence. You see the same kind of defects in diabetes and in obesity. And we have started to wonder whether there's a common insult that is at the heart of that. So I'm not going to give you a whole crash course on developmental biology because, in fact, I thought I had to, because originally the program said my lecture would start at 1:30 a.m. and I was thinking very hard, what would I do for 12 hours? And I would go to developmental biology, but thankfully that got rectified in the final version of the program. So when we look at two critical developmental processes, one is termed gastrulation. That is the formation of the three embryonic germ layers. You have the ectoderm, which will make the skin and the nervous system. You have the endoderm, which will make the gut and the organs that come off of it, such as the lung and the liver. And then you have the mesoderm, and that's basically everything else. Muscle, bone, kidney, connective tissue, blood. So that is laid out in a very early process in the embryo that happens in human pregnancies around 19 days, or it's actually starts at 19 days because it's not a process that's very short, it continues over a few days. And the mouse with a gestation time of about 19 days, it happens about 6.5 days and goes on all the way to day 12 or 13. So it's very early, right? Now the other part that I would like to draw your attention to is neurulation, the process of closing the neural tube, and I'll show you a little bit more in detail on that. The neural tube is the primitive precursor of the nervous system. It starts out from a tubular formation that comes off of a sheet of cells. That happens around 25 days, in the mouse around 8.5 days. And one of the critical points in terms of public health concerns is that those events, they're extremely critical for a successful pregnancy, happen at a time when the woman may not even know that she's pregnant. So that puts a big problem on the business of prevention, because you cannot prevent when you don't know what has happened. It becomes very difficult. It puts a light on the society here in the US, where more than half the pregnancies are not planned. So you cannot go to, if you're not planning to get pregnant, you can't go to the doctor, get your pre-conceptual vitamins and all that. And so I think it's, from a developmental biologist's point of view, even more of a reason to try and understand what actually goes wrong, so that we may have a chance to interfere in other ways than the classical clinical counseling that we have now. All right. So closure of the neural tube is shown here from a paper by John Wallingford. This is a human embryo, which starts out as a disc of two cell layers, the epiblast cell layer on top and the what will become the future endoderm at the bottom. You have a formation of a ridge, a rising of tissue, an apposition, and then a closure, that then proceeds forwards and backwards in a, as you can imagine, like a zipper-like fashion. If you cut this embryo into pieces, you'll see that where it's already closed, the tissue in question has formed a tube. Where it's open, it has several hinge points. There's a bottom hinge and a lateral hinge, and this is where the tissue moves. And we, in mouse and in human, we not just have one point where this initiates, we have several zones where that starts. It is a relatively complex process that's illustrated by the fact that in the mouse, we have about 400 genes that either contribute directly, as evidenced in the form of gene knockout phenotypes, or indirectly, as evidenced in the form of gene knockout phenotype modifiers that contribute to this process of neural tube closure. The search for a human genetic cause has been relatively unsuccessful. We think, well, you know, it's 400 genes, we've got to find something in humans, and we have very good evidence, for example, from the folate biology that we can supplement with folate and fix neural tube defects, and we should think that maybe some genes in the folate pathway actually contribute to a genetic risk, and the search has been long and hard and so far relatively unsuccessful. There are a few sequence variants, but nothing that would really stick out. And one of the reasons might be that you may actually have a multifactorial etiology. One has to keep in mind, we mouse people tend to think in terms of mouse knockouts, right? It's the be-all, end-all. If you have a knockout, that's it. Human genetics doesn't work that way. Lethal mutations are counter-selected. They do not appear in the population. In the mouse, and where I set up and decide who breeds with whom, I can keep a knockout forever. In a naturally occurring population, those alleles are going to be disappearing. So that's one thing that one has to keep in mind when we talk about mouse model systems and the human disease that we actually like to study. So how do we study diabetes in mouse model systems? We focus mostly on type 1 diabetes, meaning destruction of pancreatic beta cells, loss of insulin, hyperglycemia. We can do this in two ways. One way is to do biochemistry. There's a toxin with which we can kill pancreatic beta cells. It leads to a very rapid rise in blood sugar. It's a drug that's very unstable, so it doesn't carry forward into pregnancy, and the embryo is actually not affected by the drug itself. The neurotube defect frequency, depending on the genetic makeup of the mouse and the diet of the mouse at the time, can vary between 6 and 22%. So compared to what we see in humans, that's already a very strong model, a very high incidence of drug defect. We also have a naturally occurring model for type 1 diabetes, and that's the NOD mice. That stands for non-obese diabetic mouse. That strain of mice in females around 12 weeks of age spontaneously develops type 1 diabetes, and it's actually a pretty good model of what happens in human type 1 diabetes. It is an immune system-based destruction of pancreatic beta cells. We don't need any drugs or toxins. We basically set up the mice in the mouse and wait. The neurotube defect frequency can get up to 40%. We may have a potential complication of neurotube defect susceptibility through the genetics, and it's not very clear what the immune system impact is actually on placenta function, but these are the two models that we have available. One has to keep in mind that the most prevalent form of diabetes these days is type 2 diabetes, which is very difficult to model in mice in a way so that the mouse is still reproductively, actively reproducing, so you can get pregnancies. Most mouse models, the insulin resistance and overt hypoglycemia of type 2 diabetes comes rather late, and so that's why we've decided to stick with the type 1 model for our studies. Here we have a mouse embryo. Just to give you an idea of the scale, this is 5 millimeters here, and so we have, at that time, this one actually looks like an animal already, right? So we have a clearly defined head with an eye and ear. The jaw is not formed yet. The heart's on here. You can actually see it beating. There's the forelimb, there's the hind limb, and you have sort of this segmentation pattern here. These are somites, the precursor of the segmented mesodermal structures, such as the ribs, for example, and some of the musculature. Here's another fellow, and you may be able to see that here the integrity of the spine at this point is disrupted, and in fact this is a classical case of a spina bifida where the neural tube, so simply from here to here, from basically that point to that point, has not closed and has remained open. Mice like that will actually develop fairly normally, and you can imagine, depending on where that dysfunction is, that determines how severe the phenotype would be on an animal that's born. Because if that happens down here on the level of the hind limb, that is a lot less severe than if it happens further up the neuraxis. Because while the cell differentiation, the organization of the nervous system in that region is fairly normal, the connectivity in the end is disrupted and lead to, again, when this happens up here, it can have much more severe consequences. So we've approached this, and I'm now going back almost 10 years, originally like this, as a black box. I admit that it's not very imaginative, but at that time, what we had on information available is that it's glucose, it's ketone bodies, it's arachidonic acid, it's hypoxia, it's oxidative stress, it's glycation, it's the extracellular matrix, or the combination of any of those factors. And there were plenty of papers that from very good experiments that suggested that either one of the other is the cause of maternal diabetes causing birth defects, and of course various means of therapy have been suggested. The idea that we should do antioxidants during diabetic pregnancies has been around since the mid-80s, and it for some reason hasn't gotten any traction, hasn't worked very well yet. What it still boils down to is that a emphasis on very precise glucose control during pregnancy is actually still the best bet of how to avoid birth defects. But stepping away from the black box, what we've been trying to do is to understand two things. Number one, how come birth defects arise in diabetic pregnancies? Number two, what happens to embryonic development overall? What are the general effects of maternal diabetes that may not be related to birth defects, but to issues like epigenetic change, developmental programming, and disease susceptibility later in life? So we've been trying to tease those two things apart, and the approach that we've taken is to start looking at gene expression in embryos of diabetic pregnancies. And so I would like to lead you through 5 vignettes today that will demonstrate various aspects of the journey that we've taken. So we looked at gene expression in the embryo of the early placenta. I will show you some of our efforts towards looking at epigenomic footprints in chromatin. I would like to discuss the issue of phenotypes of incomplete penetrance. And that is something that the geneticists know, but the developmental biologists hate, because if you make a knockout and all the animals look the same, something is bad. Yet that's the reality we're going to be facing. I think I can make a very good case that we have a very good handle now on the potential mechanism of how malformations of the neural plate and neural tube arise, and we've started looking at the gut microbiome in those pregnant mice. So as I said, we decided not to dabble on the genome side. That's too hard, and in mice it's probably not fun either. My favorite sort of picture there is that everything that comes from the genome anyway has to jump through this hoop. That is the epigenome. Without that, you can have all the mutations you want. If the gene is not expressed, it's not going to matter. So we've been looking at epigenome and transcriptome, and again lately on the microbiome as modulators of phenotype and physiology. All right. Gene expression in early embryo and placenta. Then with early embryo, I'm talking about the embryos that I just showed you, 10.5 days of gestation, neural tube closed, so we can clearly see who has a phenotype, who has not. And it's more of a generalized view of what happens under conditions of maternal diabetes rather than a directed mechanistic view on the neural tube closure itself, because you would argue at 10.5 days when the neural tube is closed, it's too late to see a closure defect. But that's what we wanted to know. What happens to the embryo under these conditions? And this slide summarizes a set of experiments done with expression microarrays back in the good old days with Affymetrix 3' expression chips, GCOS software, whoever remembers that one, and differential expression analysis with a tool that actually still like to use, pulse-IRT. And what it shows you here is a comparison between placenta and embryo. These are specimen from the chemical-induced diabetes. What we see is that you see here that in placenta the up and down regulated genes are about even money, in the embryo, the majority of genes that we see as differentially expressed with certain statistical and fold change filters are downregulated. I've given you here a sort of a pie diagram idea of which pathways and which biological processes may be involved. We see a lot of chromatin transcription factors and signaling on the embryo here, and it goes more into extracellular matrix and metabolism, protein processing on the placental side. But what is striking is that we find about 2,000 genes here in the embryo. Out of those, we went through the list, we find that 388 had documented developmental phenotypes, 161 when knocked out were responsible for heart defects, 112 when knocked out were responsible for neural tube defects, and we have a clear emphasis on transcription and signal transduction. In the placenta, however, we see a much smaller footprint. I would see about 158 genes. We use a cutoff of 1.5-fold. That question came up yesterday really quick about what does it mean if we have a fold change cutoff of 1.5, and biologists usually don't like that, but I dare anybody to basically reject the 50% raise in salary with factor 1.5, and I usually would think it's nothing to sneeze at. So one can look at it in different ways. So, but what I wanted to convey with that summary of those several experiments here is that so we can see general effects. In the embryo, we do see signatures for genes that are actually involved in neural tube closure, and those genes are all changed in the direction that one would expect. They're downregulated. That's the same general direction as you see in a knockout. And one of the favorite pathways that we've been working on a little bit has been the Wnt signaling pathway. A crucial developmental signaling pathway in many instances of cell and territory specification in the embryo, and also in cell differentiation. The placental effects are more on matrix and metabolism. What we find interesting is and I'm not talking about that today because this is a genomics oriented talk anyway, but we've done histological analysis of diabetic placenta, and one of the issues that pop up is cell migration that may be impaired, and that would go along those lines of extracellular matrix genes being deregulated, and so the cells that migrate in the process of formation of the chorioalveolar placenta, they do not migrate to the right places. If you look at processes of gene regulation, the next question obviously is, well, what changes that? What changes the gene expression in those embryos from diabetic placentas? And the most parsimonious answer is, of course, it's glucose, right? Because after all, we have a systemic condition of high blood glucose, but then we started to interrogate those 2,000 genes for the presence of, for example, a carbohydrate response element. And there's nothing that sticks out. There's nothing that's really enriched. So it's not a simple explanation that it goes through the carbohydrate response element regulation and then there's that. There's another footprint in terms of glucose responsive gene regulation. It's LXR, liver X receptor. And if you look for binding sites for that next to those 2,000 genes, it doesn't work that way either. So there's no enrichment of LXR binding sites. And so nothing that would indicate that either carbohydrate response element or LXR binding sites would be responsible in the deregulation of those genes. And so that's why we decided to look towards chromatin and see whether we can find footprints of maternal diabetes exposure in the chromatin of the embryo. We can certainly find those footprints in our list of deregulated genes, and that's just a partial list because the page is too short. But what we find is a whole battery of chromatin modifiers being deregulated in expression by maternal diabetes. And a lot of these are downregulated, but one should also keep in mind that we find genes that work in opposite direction downregulated. We find histone acetyltransferases and histone deacetylases downregulated. We find histone methyltransferases and demethylases transferated. Now, these classes of enzymes typically work in opposite directions. So if you acetylate a locus, you usually have higher gene transcription. If you deacetylate, you have lower. If you methylate the locus, you have lower transcription. If you demethylate it, then you have a chance for higher transcription. So it's not a very simple and a unified change that we see just on the basis of gene expression. So we underwent a scheme of looking at chromatin from early embryos with 5 to 7 of those little mesodermal blocks that I showed you earlier. And the rationale for going after histone acetylation in this case is as follows. So high glucose is known to be correlated with high content of nuclear acetyl-CoA, meaning the acetyl group that's actually being transferred onto the histone moieties comes from acetyl-CoA, and there's actually a nuclear pathway through the acetate citrate lyase gene that produces acetyl-CoA directly in the nucleus. You don't have to go through other parts of the cell. In fact, acetate citrate lyase is in fact a neural tube defect gene if you knock it out. So there's a chance that there's a direct effect of this excess glucose on histone acetylation patterns in the embryo. And we wanted to focus on one particular mark in this case, and that is the lysins 27 of histone 3. And the reason for that is this residue has a bivalent functionality. If it's trimethylated, it's correlated with repression of transcription. If it's acetylated, it's correlated with enhancer function, meaning activation of transcription. So that is a critical regulatory residue that can actually do both things. It's not like if you modify it one way, it does something, and if you do not modify it, it does nothing. There is always a balance between either methylation or acetylation on that residue. Next part is that what we found actually our interest is centered on a gene called EP300, and that in fact encodes the acetyltransferase that modulates modulates the K27 residue. EP300 is a neural tube defect genus. If you knock it out, you get neural tube defects. If you get open neural tube or exencephaly, that means the nervous system in the region where the brain forms is open and not closed. So we used whole embryos in this case, and this is before you get weekly advertisement from Diogen Node or Activity that you can make chromatin libraries from 500 cells and things like that. So it was a little bit more of a struggle back then. And just to go through that scheme, so we used whole embryos at 5 to 7 somite pairs, about 30 of them or so. We went with enzymatic sharing of the chromatin. Our size classes actually correspond to DNA that we would expect to be wrapped around 1, 2, or 3 nucleosomes. The technique for that is, for the sequencing itself, is with the applied biosystem solid 5500 XL technology, and we did the 35 base pair sequencing because, in our experience, that is the sweet spot of that particular instrument. We have about 30 million uniquely aligned reads, and for this particular instance, we allow zero mismatch. So these are 35 base pair zero mismatch alignments. We have an instantiation of Galaxy at Pennington, and that has been our wrapper to conduct these analyses. And basically did the alignment with bowtie, run a program to recognize histone modification islands, that's called SICER. And then you run that same program again to compare the location of those histone modification islands in order to derive what we call histone modification features. So this is sort of, if you follow that, this is that pathway down here that describes the H3K27 acetylation analysis. So, in fact, we do find changes of acetylation patterns on H3K27 throughout the genome. We find about 11,000 of those islands we find that about 2,000 of them were upregulated under conditions of maternal diabetes or had higher recovery in the ChIP-seq. And about 3,600 are down, meaning there's lower recovery in the ChIP-seq compared to input chromatin. And you can make nice volcano plots plotting the statistical significance of the fold change. What I found interesting is when we started to look a little bit more closely at where those elements are that we see changed by maternal diabetes. And this is one of those things where the ChIP-seq patterns and histone patterns carry a big question mark in my mind, because particularly in developmental situations, you never know how far away from the gene a epigenomic modification can be and yet still affect the gene itself. And the worst case scenario that I know of is, for example, the sonic hedgehog gene. There's one known transcriptional enhancer that's 1 million base pairs away. That if you were to look at sonic hedgehog regulation with the classical concept of I'm going to look at 1,500 base pairs upstream of the transcription start because that's a promoter of the regulatory elements, you will never find true biology of sonic hedgehog regulation because that enhancer is far, far away. And in fact, it wasn't even found by classical reporter gene studies where you take a piece of DNA upstream and test it with a reporter gene. This was found through actually a human genetics approach where there was a breakpoint in the chromosome. All right, so we made different size windows. So we allowed 50,000 base pairs up or down, 100, 200, up to 350, and then counted how many of our neural tube defect genes, because those are the ones we're really interested in, would have an element that's downregulated by maternal diabetes, which would go along with the downregulation of transcription that we see because this is an enhancer element. And we find those, and in order to test whether that has any kind of meaning, we performed a random draw strategy where we draw randomly gene names out of the list of the 26,000 rep-seq genes in this particular case, and ask the question, do they have an element that's in the same size range? What I'm showing you here is this is the mean from all these random draws. This is the highest value, this is the lowest value. And then you can do a Fisher's exact test to basically query whether that enrichment has any kind of statistical significance. And it turns out for neural tube defect genes, there is a clear statistical significance for the elements that are downregulated. When we do the same thing for elements that are upregulated for diabetes, there's no such thing. In fact, the curve for my favorite set of genes is smack dab in the envelope drawn up by the random draw method. And so the random draw, by the way, I should say, this is bootstrapped and commuted in order to arrive at a quantum p-value from that. So the presence of those histone acetylation features where maternal diabetes reduces the overall enrichment of acetylation could be a potential mechanism to explain how the downregulation of a lot of those genes on the question works. But this all, the question though is, what does it really mean in terms of biology? And here's where I think the power of the genomics has not yet sort of meshed in a very good way with the functional testing, because when we find a signal, for example, here's the representation of the EP300 gene, and in fact we find two spots here where there is histone 3K27 acetylation, but it's down in diabetes. And in fact, this one actually overlaps with the previously described enhancer of the gene, so we are pretty confident that we're actually hitting the right notes here. But what does that really mean if this is down? Does it really mean that EP300 is transcribed at lower rate? And we find similar patterns to other genes of interest. Here's a nodal gene, that's a gastrulation gene, we'll come back to that. There's the mark. Here's the T gene, also called brachyury. There's a nice signal here, and so on. There's FNA1, and the signal here. Or in TBX6, there's a chunk, histone feature right in the next gene, but right upstream of the gene. So we find interesting and compelling chromatin signals next to genes that, that we find really important. But how do we test the functional significance? I'm not going to show you because we're just starting on this, but the true mechanistic test for that is actually epigenomic editing. We need to go into the genome and modify those epigenomic signatures in order to really test what is the impact if that area does no longer carry K27 acetylation. Does that really mean anything for the gene itself? So in that sense, the ChIP-seq data were actually very encouraging because they show us a lot of facets where we find that the crucial developmental regulators are affected, but the mechanistic test by itself will unfortunately not come through genomic trickery, I should say, but to old-fashioned developmental genetics, where one has to go into mouse stem cells or into transgenic mice and engineer those areas so that one can functionally test of how this actually works under biological conditions. So the next part is where I'd like to show you something to think about what it means if you have certain kind of phenotypic constellations. And I'd like to show you the solution first, because this is what we all strive for, right, when we do differential gene expression. What we want is, we want a nice difference in mean, we want a very low degree of variability, so we get good p-values, and we can write that in our papers, and the reviewers are happy, and the NIH grant reviewers are happy, and it's all great and nice. And we've done that, okay? And cool. The problem, though, is this. See that? That's not here, right? So what we have here is a phenotype of incomplete penetrance, right? Under normal conditions, everybody is above this imaginary line, and everybody here is normal. Under the diabetic exposure conditions, what we have is, yes, we have neurotype defect phenotypes in our model, but the majority of embryos in this case are actually quite all right. Okay? They have no neurotype defect phenotype, and maybe they have some developmental programming, but if you would let them come to term, they would walk away and be a regular mouse. So, is that really the solution to this problem? So we have a phenotype of low penetrance, and we're trying to contrast that with genes that have a high correlation in their expression changes with maternal diabetes. And so the question that we started to ask ourselves is, how can a gene that's changed all the time cause a phenotype that you observe only part of the time in the embryo? Okay? So if those genes were really responsible, should we have a high penetrance of the phenotype? Shouldn't everybody who has this downregulation of gene X have that neurotype defect phenotype? And so that sort of started an itch that we're still trying to scratch. And the other one is this idea with the systemic condition alone. When everybody's exposed, keep in mind that we're now talking about rodent pregnancies. We have about 8 embryos per liter. Everybody sees the same degree of blood sugar that the mother has. How does that condition, that exposure to high blood sugar, just by itself, how would that explain that we have, in a litter of 8, we have 1 or 2 neurotype defects, right? So things are, and there's a disturbance in the force. Things are not aligned, and I'll try to show you how we try to take advantage of that. So when you look at phenotypes of reduced or incomplete penetrance, this is nothing new that we invented. It goes back to skillful observations by Grunenberg in '52 and seminal observations by Clark Fraser in 1957. Clark Fraser is a teratologist extraordinaire who unfortunately passed away not too long ago. Here's a direct quote from Grunenberg, but the way you can think about it is as follows: that on a susceptibility scale, under normal conditions, there's got to be this bell curve, and a very, very tiny fraction may cross a threshold, the threshold that goes from between normal and an affected outcome. And when you have a teratogenic situation, we can think about it of what it might do in two ways. It could be that it shifts, that heterogenic modifier will shift the mean of the whole population over. So if you go from this curve to that curve, and the shape of the curve is the same, but now since you're further over to the right, a lot more individuals cross that threshold, and you have an increase in the affected population. What you could also think about is if you change the shape of this distribution. What if you just increase the variability and the spread? Instead, you would have the same effect, that you no longer have this short bell curve, but it's still a bell curve, but flatter, and you still have this increase in the number of affected individuals. And in order to address that, we started to look at our gene expression experiments in a different way. And the simplest way of how to do this, or what was actually shown on this one here. So what we did here is we looked at microarray data, and we just plotted the standard deviation that you see for each gene in the normal case, which is on the x-axis here, and in the diabetic case on the y-axis. And if all things are even, what you would expect is that, and here's the diagonal, what you expect is a data cloud roughly aligned with the diagonal. Some here, some there, depending on how precise we can measure. But overall, you would say that the data cloud should be aligned here. And what you can see here is that the whole data cloud is shifted towards the diabetic side. Okay. Indicating that we would generally have higher standard deviation in the diabetic exposure paradigm than in the normal situation. And that is with 20,000 genes on the array. So that's not selected genes, that's just taking everybody. A slightly different presentation is when we turn this into a coefficient of variation, log transform, we plot the two curves, and you can see now we have quasi-normally distributed curves. You can do a t-test. P-values are fabulous, 10 to the minus 300, it's great. But that's cheating with 20,000 samples. Everybody can get the p-values. Kolmogorov-Smirnov test still says it's statistically significant. So is it okay? Fine, it's the embryo. Well, lo and behold, the placenta looks the same. Same deal, the diabetic curve is shifted further over to the right, indicating a higher degree of variability in general. So we looked on the web and decided we need to do something where it can't be our fault. So we didn't make the RNA, we didn't make the sample, we didn't select the sample, we didn't make the RNA, we didn't hybridize the arrays, we didn't do the evaluation. This gene expression pattern on glomeruli in human diabetic kidneys, and I was very pleased to see that we see the same shift of the diabetic samples further over to the right, indicating a higher degree of expression variability. A couple of years later, we did microarrays in the non-obese diabetic strain of mice. This is actually a series where we did pooled samples. So there's where an n equals 4, and each single sample consisted of a pool of 4 samples. And again, we see the variability shifted in the same way as we see in all the other paradigms that we've done. So you can still blame us for ruining it in the lab for this, but not for this one. We then started to think that we may actually turn this increase in variability, which by all intents and purposes you really didn't consider as bad and terrible, and you have to adjust for it and all that. And we started to look at that, the variability itself, as an experimental parameter. So what I show you here on the top is classical hierarchical clustering, Euclidean distance, actually done in Mike Eisen's cluster 3, which was written in 1996, I believe, but math is the same, just more old-style program. But it shows you 2 heat maps here. One is from the chemical model of diabetes induction, one is from the naturally occurring diabetes. And what we can very easily do is, by hierarchical clustering, distinguish between the control individuals and the diabetes-exposed individuals. In each case, that's very easy. There's a nice deep branching pattern here and here. So it's very simple to determine who's been exposed to diabetes and who's not. Now, in both experiments, we had included a few samples that actually had a neural tube defect. So these are embryos that had a morphogenetic deficiency. They had an open neural tube. And no matter what we tried, on the basis of just differential gene expression, we were not able to cluster out the individuals that had the morphogenetic defect. We could only separate between control and diabetes, but not between diabetes exposed normal versus diabetes exposed malformed. However, when we started to do the same experiment based on variability from the mean of the normals, on the variability of gene expression, not on the overall distance from of how the means are different, but the variability of each individual in this group, we can cluster out the neural tube defects very easily and very powerfully. And it's the same hierarchical clustering. We didn't change the parameters. It's still a Euclidean distance paradigm. And we can do it in the chemical model of diabetes and in the naturally occurring model of diabetes, that the expression variability itself tells us something that we were not able to extract with the pure view of looking at differential gene expression. And so I think we may have to look in these cases where we have these type of phenotypes, we may have to include a few more conditions in our analysis paradigm. And I'm the first one to admit that it becomes relatively messy, because that is easy to analyze, right? We got programs for that, t-test, Bayesian, moderated or not, and a few other things. But who is to say that if this is the susceptibility condition, then a gene that shows the expression pattern like this may have two of those individuals in that realm where it crosses, where on the wrong side of the threshold. And so we've looked at things like that and started to do pathway analysis based on these patterns. But overall, I think we need to think about what is our phenotype like before we basically strap on a model that works so well in many other circumstances. But in this case, I think it's when we have this situation here, we need to account for that kind of population in our gene expression paradigm and make sense of it. And so that's the picture I would like to leave you with. So classical differential gene expression may be insufficient to explain those phenotypes of incomplete penetrance. And I think, at least from our array studies, we could make a cause that this model of the unimodal change, where we do not invoke other parameters except for variability controls, could account for those phenotypes that reoccur. We can't rule out completely that we could have a biphasic, bimodal action. And for example, in the case of gene knockouts or so, you would probably argue that it's more like this. And in fact, a lot of the gene knockouts, when you read the first paper in mouse genetics journals and developmental biology journals, you see the mention of neural tube defects, and everybody thinks this is a neural tube defect gene. And when you read the third or fourth paper on the same gene, suddenly there's going to be it's only in 7% of the cases, right? So even in mouse knockouts, it's not always as clear cut that the whole population shifts over to the phenotypic side. All right. So far, maternal diabetes exposure, epigenome effects, gene expression effects, but what about birth defect phenotypes per se? So we looked at embryos at the time when the neural tube closes, and here is a picture of that. And you can see that we're not much smaller now. This is about 2 millimeters here. What you see is here is the midbrain. The forebrain is further down that you can't see. The hindbrain's here. Here's a closed neural tube. You see those little blocks of tissue. These are the somites, the segmented mesodermal areas. And back here, there's still open neural plate. Okay, that's about 8.5 days of development. So this is how it looks in a normal situation. Now we looked at embryos from the NOD strain, and you can see that there's a few things that's not right. There's something growing out of the middle of the neural plate. Some very big, like this one here, some voluminous, some smaller. On a cross section on this one here. So this is an area where it's not affected, and you can see that there's something in the middle there. And so we thought this is our avenue to look into the neural tube closure process from a slightly different angle. That is a phenotype that has not been described before, and the reason probably is because these things are very small and not that easy to make anymore, and not that easy to dissect. So we did some imaging. I guess that counts as computational biology. So we did confocal imaging with two-photon microscopy, did three-dimensional reconstructions of the image of the whole embryo from stacks. That looks like this. Again, here's the forebrain, here's the heart, and here's this protrusion that grows out of the middle of the hindbrain. That is not the tail, that actually is the connection that will make the placenta. All right? And then on a 3D reconstructed image, we can cut it any way we want. And what you can see here is that this protrusion is actually contiguous with the nervous tissue, but it has sort of this loosely, slightly not so densely stained compartment in the middle. Similar picture here, another sample of that, and you can see that the outer layer is densely stained, the middle part is not so densely stained. And we wanted to use this kind of phenotype now to understand what's going on, what in the diabetes exposure does do that, and what exactly happens to the embryo. So we decided we needed to be more sophisticated than the plain microarray approach. And what I'm going to tell you now is an application of next generation sequencing, specifically the 3' expression tag sequencing, or SAGE or super SAGE. Starting with material where we dissected this little protrusion out from the embryo, made RNA from it, and keep in mind this is 2 millimeters, so this is what, 30 microns, something like that? 50, okay. And we needed a tissue to compare this to in terms of to have a good reference to understand what actually happened in this teratogenic tissue. So we took an embryo like this, embedded it, cut slices from it, and used those same slices to cut out the open neural plate with the laser. And it looks like this. So we start out with an open neural plate, cut that out, and that's what we get. And so we compared the transcription profile from this compared to that. Overall, it was a 4 versus 3 setup, but we didn't have any more. We start with 50 nanograms of total RNA here, 7 nanograms from the slices. This is about 2,000 cells overall. And again, this was a little bit before everybody started to know how to do single cell transcriptomics. So we were actually fairly happy that this worked successfully. 8 million reads per sample, 15,000 genes, 10 million over here, 10,000 genes. And that's because those two things are substantially different. The informatics pipeline is a program called Solid Sage that we inherited from Applied Biosystems and modified and embedded in the Galaxy wrapper. That gives us a count table that we then analyze with a utility called DSeq. And this is the old version. Right now we're at a much higher number. Cutoffs at false discovery rate of 0.1 and the false discovery 1.6-fold. That gives us 800 genes that differentially represented between the two tissues. 571 are in those protrusions, or enriched in those protrusions, and we had 90% validation rate by quantitative PCR. And just to give you an idea, everything that's about 50 counts we could measure by quantitative PCR in this situation. Did our pathway analysis, and I have to say this was one of the moments where it was really fun doing this kind of work, because I happened to be the one who saw those protrusions for the first time in our lab under the microscope. I also happened to be the one that needed to do the dissection, and so that's why I wanted to be the one who gets to look at the sequencing data first. Yeah, but that's part of the fun. We're laughing, but that was really the fun part, and part of. I don't know, I've still been fascinated, and I still have that moment in my mind when I did the pathway analysis, and we find that top category is induction of mesoderm, okay? So we find that in this material that grows out of the neural plate, which should be neuroectoderm, we find mesoderm. That's the filler tissue between endoderm and ectoderm. It's not supposed to be there. And talking about interactive graphics, Dr. Jack that we heard yesterday, I wished it would have been like this because all its annotations that you see here are done by hand. And Susan Newman, who does a lot of these things for me in the core, has much better tools, but then back then I wanted to do it myself, and so I painfully draw circles around it. So it would be cool to, but Jump Genomics has things like that, but I've been too boneheaded to move from that. But it was really fun, and I have to say this is it's still been a source of fascination and fun to see that that biological answer that popped out of that sequencing experiment. When we started to look a little bit deeper, we find that actually 85 of the genes had published expression patterns, localization by histology in the embryo, and 33 of them were actually exclusive to mesoderm. And I mentioned two words here, node and primitive streak. These are crucial tissues that have inductive function during the embryo. These are the tissues that make gastrulation. And I told you gastrulation is an important part of your life, right? Without that, you're not going to exist. But what was more important to me is that we saw not just single marker genes, like some of these may be, but we saw induction of whole gene regulatory networks. So we see the transcription factor T here. We find 67 of these known targets in that same list of 507. Okay? One of them is CDX2. That's another transcription factor. We find 150 downstream targets of CDX2 in this mix. So that gave us confidence that this wasn't just a few marker genes that popped up in the right fashion and it was an accident. This was a coordinated induction of regulatory networks that have clear developmental significance, and that's why I've been so excited ever since I saw that first IPA analysis that we did. So why am I so excited? Because it's clear to us that maternal diabetes affects the neural tube closure by actually acting on a process that happens much earlier, gastrulation. And it's this ectopic generation, this out-of-place generation of mesodermal cells that we think is the cause for this morphogenetic defect. In terms of what else is in those gene regulatory networks, we see actually three big footprints besides the pure transcription factor networks. We see one network that's known here, that one. The question is, what comes first? Is it, do we make more mesoderm from scratch? Do we keep making mesoderm in the place where it's happening when we make it too long? Or does the mesoderm simply stick and it can't walk away? And these three signatures are all there. And with keep in mind, we've done a simple time point. So we don't have a temporal sequence. And in fact, that is one of the specific aims of my new grant, to establish a temporal sequence to find out who comes first. And so we'll be doing a lot more of this kind of sequencing. The interesting message in general was this refined functional genomics, this going down to a much more sophisticated preparation of the sample. That was what really opened the door for us to move on forward. And now, as part of the lab has actually stepped away from the nucleic acid biology in this one, and the whole finding allowed us actually to sculpture a mesoderm bioassay that we can now move away from gene expression, but actually test on a mechanistic level. Why is it that those mesodermal cells, A, appear, and B, why don't they go where they're supposed to be going? And particularly, what we know now is, why are they moving so slow? And this is where, many slides ago, I showed you this issue with the placenta, that we have a situation that cell migration is not working right in the placenta, and it's very clear to us now, with the first results of the bioassay here, that the cells are not moving right, they're slow. And so our next analysis tool is actually going to be bioimaging. And I want to close with a very short vignette on the gut microbiome in pregnant diabetic mice. This is happened sort of with the encouragement of Chris Taylor. So we compared cecal contents from pregnant mice at 8.5 days of gestation. These were the same animals from which we prepared the embryos. And just for from a technical perspective, we did variable region 3, variable region 4 paired-read sequencing on an Illumina MySeq. Made our overlap and quality scores in a context with software called Mother, and then sort of started this tiptoe between different software pieces where we felt we get the best result. So in order to do the alignment, we went back to a software called uSearch, removed chimerics with uChime, got our operational taxonomically unique count table, again with uSearch, and this is where I'm happy if I have a count table. I know I can do DSeq, I can treat it as any other kind of differential representation. And I'm not a microbiologist, but I felt we need to do the rest of the analysis because it's just state of the art. But my main thing is I want to know who's different and by how much and with what scientific significance. So in order to do the taxonomic assignment analysis of communities and diversity, we start to use Mother again, which now transitioned to phyloseq. But again, so my main belief is always in the statistical test at the end. And here's just a snapshot of this analysis that maternal diabetes actually leads to a reduction in community diversity. So the microbiome has a reduced composition of different species. So the alpha diversity is different diversity between the groups with the various tests comes out as statistically significant. And what you see in blue here in this space are the microbiome points from the normal glycemic mice. In red, purplish are the ones from the diabetic mice. And what we can see when we rotate this cube is that the normal ones form a tight unit in space of these principal coordinates, whereas the diabetics form a cloud around it. So again, we have this issue where we see a higher degree of variability. What was funny is to see this guy up here. You can see that there's a blue sample right to a pink one, and it turns out that is a case of which is gestational diabetes. This is a mouse where we had perfectly normal blood sugar at the moment we set the mouse up for mating, but when we actually just dissected the embryo, it was hyperglycemic. We see a couple of the usual suspects, I should say. There's various lactobacilli that are severely decreased in the diabetes-associated microbiome. We see an increase in clostridioides throughout the diabetic microbiome. Now, I'm not a microbiologist, and so I have very little idea of, except that Clostridia are relatively bad, but I don't have any intention of going into the actual microbiology. What I'm starting to get more interested is, again, more, as I've listed down here, I'm more interested in the functional significance of what happens when the microbiome is actually changed. And the way we're trying to address that is to a microbiome transplantation. So we have now a protocol, and again, this is in collaboration with Chris and his folks in New Orleans. We have a protocol in Pennington established where we can deplete mice with antibiotics, transplant the microbiome, and then measure physiological parameters. And that's mostly due to the efforts of Dr. Amadoro Bruce-Keller. So we see significant differences. What I should say is, if you followed the research in obesity a little bit for a while, it was the big difference was that you have a much higher degree of firmicutes versus bacteroidetes in microbiome from obese people. We do not see wholesale changes like that. We see changes within the firmicutes, we see changes within the bacteroides, but not a complete shift over on the grand scale. And again, the reduced replication of certain species of Lactobacillus and Akkermansia municiphilia, I think, which is generally been thought of as a good guy. So, I'd like to leave you with the conclusions that we have general effects on the microbiome, on gene expression in the embryo, and the main signature that I'm excited about is this. That I think that's my interpretation of this variability business I tried to show you, is that maternal diabetes leads to a reduction of the precision, of the regulatory precision. That is what gets lost in the embryo. That means a lot of them can still make it and walk around fine, but I think that is one major point I would like you to take home. And the other part is, so that my fun part is this specific target of gastrulation that actually precedes the neurotube closure, which is where the overt phenotype is light. And the more important part in the end is that I've talked about heart defects very briefly, but keep in mind the heart is formed from mesoderm, and in fact the heart mesoderm is the first one to migrate. And so with this gastrulation model, we can explain the other birth defects in diabetic pregnancies. And I'd like to close with an acknowledgement of people in the lab. This has been a long-standing collaboration with my co-conspirator, Claudia Koppen, who has a chance to be here today. Jackie McGowan has been doing a lot of mouse work. Claudia Kruger is a molecular biologist par excellence, has been making the libraries for the sequencing. Niels Herren has been a very enthusiastic, I don't know exactly what he was, undergraduate, like student, but a lot of fun with that. I'd like to acknowledge David Burke in the bioimaging, because without him we wouldn't have been able to characterize that phenotype. Without folks in the genomics course, Susan Newman, Richard Carmouche, and Diana Holmes, nothing of that would have happened, and nothing that would have happened without funding from the NIH through our Munster, Claudia, and myself, and from funding to the NORC and the COBRA. And with that, thank you for your attention, and be happy to take questions.
This was all in the mouse model, so, and I know that at the beginning you said that there's about 400 genes that change in the mouse, that there was no detection that had been done with the defects in it. Can you make some analogies from these 5 points that you found into the human, what you think is happening there?
I think the variability is something that I would think would translate over. The gastrulation phenotype is a little more difficult. So I've seen some pictures of human embryos with anencephaly, meaning that the brain is completely missing, where there's sort of an amorphous mass left over in the middle of the middle of the brain. And I'm biased because I've seen the other things on the scope, and I'm thinking that may be a remnant of such a protrusion that's there. There is not a very good collection of human imaging data where we would look at ultrasound images of embryos at just different gestational levels. And for example, one of the reasons is that it's, nobody thinks about doing that on a regular basis. But the studies have increased in recent years, and it has changed our perception a little bit. In the old days, it was thought that human neurotube defects are only spina bifida. Well, those are the ones that are survivable. An anencephaly, if you don't have a brain, is not survivable. So you do not find those phenotypes. In the mouse, in fact, you have more anterior defects in the mouse, in the brain, and not so much spina bifida, except for this model where we actually see a fair amount. But it would be nice to have more imaging studies in humans. The problem there is you talk about very, very small changes that are difficult to image even in the mouse embryo. And so I'm not so sure how we could get at data like that experimentally verify directly. Can you look at abortion material? I don't think so. I don't think that's a good way of doing business. to look at material that's obviously either has been destroyed or has been defective in the first place. That doesn't help us much in assessing is this a process that could go on. But as far as the mouse model goes, I think we've made a significant step forward in looking at this process. And we have some new studies in the pipeline where we can actually link oxidative stress into exactly that process, into the cell migration process at gastrulation. So I think this provides us an opportunity to delve deeper into the system than ever before. And in my mind, it was made possible by the sequencing approach on very, very small amounts of tissue. And I should say, given all the microarrays that we've done, on the sequence, we see things that we do not see on the microarray. We know they're there because we can see them by qPCR, but so the sensitivity and also the selectivity through the laser microdissection, I think that was the key point to move forward.
Thank you.
And I should encourage everybody, it's not that hard looking at data. You know, just do it. Thanks for your attention.